Progenitor cells are busy cells. Progenitor cells for the nervous system are busy. They're in the central nervous system alone. We're not even going to talk about the peripheral nervous system in this regard, but in the central nervous system alone, we're looking at trying to get 170 billion cells made. And so there has to be a lot of division. And this happens because progenitor cells have two ways of dividing. In one way, which happens a lot early on, they have a symmetric division so that they form two more progenitor cells. The other type of division for a progenitor cell is asymmetric, where one progenitor cell is born and one terminally differentiated neuron or glial cell is born. So these divisions, these are divisions of very potent cells. Now, if you looked at an egg, it would be called pluripotent. It can become anything. But the progenitor cells in the nervous system are multipotent. They can't become a kidney cell, and they can't become a lung cell. They can become a neuron or a glial cell. That's what they've got available, or another progenitor cell. So, we've limited we've narrowed the possibilities. These are types of stem cells, but they are limited. They are not omnipotent. They are not pluripotent, as it's called, but they are multipotent. They become neurons or glial cells. Now, the rodent, in the adult rodent, the adult rat or mouse, there are a lot of these pluripotent cells. So if there's injury, you can get new neurons. In the adult human, there are a very small number. There are two sources of pluripotent cells. One is in the dentate gyrus, and the other is in the subventricular zone, which migrates out into the olfactory bulbs. That rostral stream is dried up in the adult human. There are pluripotent cells, progenitor cells that can make neurons in the dentate gyrus. But for the most part, if you lose neurons, you cannot replace them as an adult human. You might be able to replace some of them if you were an adult rat or mouse, but if you're an adult human, you are not going to get neurons back. You're not going to be able to replenish the number of neurons once they're gone. Now, there are progenitor cells that can still make glia. So we can make new glia as adult humans. And that's very important when we have diseases such as multiple sclerosis, which is demyelinating. There might be, demyelination can happen for a variety of reasons, but if it happens because of a problem with the oligodendrocyte, you might be able to get back some oligodendrocytes because of this ability to replenish glial cells that is greater than our inability to replenish neurons. Now, I talk about the adult. There is a human that has a lot of progenitor cells, and that is a human that's below the age of about 5 years old. And where are these progenitor cells? They're back in the hindbrain. They're making this structure here. This structure, the cerebellum, part of the hindbrain, develops from a place called the rhombic lip. And roughly half of the neurons in the central nervous system, so around 40-some billion, are contained within the cerebellum. This is chock full with one type of cell that makes up about half of all the cells in the brain, all the neurons in the brain. These cells are only starting to populate the cerebellum at the time of birth, and they continue to divide. And this will become important later when we talk about types of brain tumors. Okay. Now, lately, the political and ethical debates over stem cells have diminished a bit. And in part, that the temperature in those discussions has lessened because we have a different alternative available now. And these are called induced pluripotent cells. These induced pluripotent cells are an incredibly exciting development where you can take simply skin cells or some epithelial cell from a person, take a biopsy, so you take out those cells from the person, put it into a dish, put on specific what are called transcription factors to get that cell, that terminally differentiated cell, skin cell, to de-differentiate, go back to eggland, okay? Become a pluripotent cell. And then you put on a different set of transcription factors and bring that pluripotent cell up to become a neuron. Now imagine if you can do this for an individual who has, say, an inherited form of Parkinson's disease. So now you know what's the mutation in this person's that is causing this person's disease. Not only can you figure out what the individual's genetic error is, but you can throw drugs at these cells that are going to be just analogous to the cells that the person actually has in their body. So that enables you now to test out drugs, see are they likely to work in this individual or not. And this is a really, really exciting area of research, and an exciting possibility for greater and improved clinical care in the future. Great. So now we're going to talk about, we're going to delve into the difference between a peripheral neuron and a central neuron.